Hey everybody, we are looking at section 19.2, which is modern evolutionary classification. So what we're going to do here is kind of build upon what was discussed in section 19.1, but also discuss and talk about a different type of classification system. So something that doesn't follow with Linnaeus's taxonomic model with the domain, kingdom, phylum, classes, and so on. This is going to be looking at classification based on evolutionary relationships, all right, and ancestral relationships. So, in other words, using some of those vestigial structures and homologous structures to help us identify relationships and place organisms into categories based on when they developed in terms of like time in the past, right? So, as always, make sure you get the title here. And then for this section, we've got 4 objectives. So we're going to look at why scientists do evolutionary classification, what the purpose is. We're going to talk about these things called cladograms and cladistics. We're going to look at DNA sequences and how those can be used as part of classification. And then kind of relate this all back to Linnaeus's model and what this tells us about his, what we would call a tree of life, right? These domains and kingdoms that we talked about last time. So when we look at evolutionary classification, like we are in this section, let me move myself up here a little bit. The goal of this is to group species according to their evolutionary history. So in other words, looking at where they originated from, right? What are their ancestors and how is it that these species have come to differ from those ancestors over time, right? What is it that was maybe selected for? So in order to classify species in this manner, we have to use lots of different types of evidence. DNA evidence can certainly be used, so that's molecular data. Fossil records, structural similarities, like I mentioned, homologous and vestigial structures can be used for this. But remember, we don't use analogous structures to infer any type of evolutionary relationships. It's strictly going to be those homologous structures and vestigial structures. So when we talk about evolutionary histories, there's a term that biologists use, which is phylogeny. So phylogeny is a vocab term. That's why it's shown in bold there. These are representative of the kind of path to creating different types of organisms and different types of species. So phylogenies, for all intents and purposes, kind of resemble family trees. And this is an example of a phylogeny for different groups of vertebrates, so different organisms that have a spine, right? They have a backbone. And you can see we're starting with the oldest, most ancient species down here, which are jawless fish. And then as we go up this little chain all the way up to mammals, we see that we become more and more complex. And so the idea is that these 2 organisms down here, lampreys and hagfishes, are going to be more related to each other than they are to something like sharks or sturgeons and so on. So in general, the more that they share in these types of diagrams, the more closely related they intend to be, or they probably are, in terms of their evolution. And these smaller branches are indicating that you've got, some shared histories there in the background. Okay, so this is again a phylogeny. This differs from Mendel's work in that we're not dealing with anything in terms of levels of classification. We're just looking at really these changes over time. Okay, so when we talk about evolutionary classification, the most commonly used system is called cladistics. And this is a whole field of research, a field of study that's dedicated to creating phylogenies. Now, this isn't the only method that we can use to create these evolutionary trees, but it is probably the most commonly used. And so the idea with cladistics involves classifying organisms based on common ancestry and or shared characteristics and gene sequences. So those evidences we talked about on the last slide are what cladistics uses to organize information in species in diagrams that looks like the one you see right here. So this diagram is called a cladogram. And I can see I have several different vertebrate species here, right? I've got turtles, horses, wolves, leopards, domestic cats. And they're all kind of branching off of this original line, right? So this original line is going to represent kind of the process and the pathway of time in a way. And then these, what we call these little outgroups that are kind of coming off of that, represent significant changes that created and led to the formation of these different species. And then here you can see these are some of the characteristics that would be true of organisms in that group. So for instance, all of the organisms here shown in purple have hair. right? So they all fit into class Mammalia. Turtles do not. So turtles are called the outgroup here because they don't fit within that major category. But everything else, horses, wolves, leopards, and cats all have hair. They all fit into Linnaeus's class Mammalia. Anything that's eating meat primarily or other animals that are carnivorous would fit in the categories to the right here. So wolves, leopards, and cats are all carnivorous, so they would fit there. They're in order Carnivora. Leopards here are part of family Felidae along with domestic cats because they have retractable claws, which wolves, horses, and turtles certainly do not. And then domestic cats have the ability to actually purr, which leopards do not, and so they fit in this final category here. So you can see as you go down this line, right, we're becoming more and more specific in terms of the traits we need to be seeing. What they share in common is represented by these little hashes here coming off of that original line. Okay, so when we think about these cladograms, there's lots of different terms that apply to what we're seeing. So again, here's the definition for cladogram, a diagram that shows how species could be related to each other evolutionarily, right, through common ancestors. So the traits that I talked about on the last example are what we call derived characters, and these are characteristics that are derived, you can think about them this way, from a most recent shared common ancestor. So they're used to identify relationships and are represented on cladograms as these little hashtags and these little, not hashtags, but hashes coming off of this original branch, okay? In general, again, the more closely related two species are, the more derived characters they would actually share. And so if we look at this example, we're going from animals of all sorts of different types. We have hagfish, perch, salamanders, lizards, pigeons, mice, and chimps. And so if we think about the mouse and the chimp and how similar they are, well, they're going to share all of these things in common, right? They have jaws, they have lungs, they have claws or nails, they have fur and mammary glands, right? There's something that distinguishes these two here, but for the most part, they're going to share a lot of things in common because they're pretty closely related. Same with lizards and birds here, right? They're closely related in terms of they all have jaws, lungs, claws, or nails, but then birds are distinguished because they have feathers. Okay, and in this case, they're representing that derived character here on this actual branch because that's not something we see in mice and chimps. Okay, so when you see this terminology, and this may be something to jot down just to help jog your memory about what all these words mean, but derived character, again, are those traits that are shared by some species but not others, and those are written off to the side, branching off of this original line. The earliest common ancestors off to the left, the very kind of bottom left, or I should say bottom left, okay? The clade is the group of species that share a common ancestor, and so actually these are all clades, right? We could consider that this would be a whole clade because they all have backbones. We could say all those with a jaw are part of another clade. You could say the mice and the chimp are part of one, but it's how we're grouping these species together. And then a node is where we have these branches that are actually splitting from one another. So each of these positions here, where we have a branch coming off and up and to the left, that's considered a node, okay? So you can have a ton of nodes represented on these, or you can just have a few. It kind of depends on how you set it up. So in looking at these cladograms, the way that we diagram these and draw these out is to consider those ancestral relationships. But anytime we have a node like this, that represents a splitting event in terms of evolutionary history. So we have, let's see here, a splitting event here that would occur involving species number one or group number one. We have another splitting event here with number two, and then another splitting event here that occurs between 3 and 4. All of those splitting events are represented here. And so again, anytime you see the branches kind of diverge and separate, that's where you have those splitting events. But again, those common ancestors are always going to be graphed down here towards the left and towards the bottom of the diagram. Okay, so if we talk again about what are examples of some derived characters, right? These are traits that these species share in common. If we compare lions to coyotes, well, they're going to share a lot of things in common, right? They both have backbones. They're both considered mammals. They both have hair. They're both warm-blooded, right? And they also are in the group Carnivora also. So they share several things in common. And if we were to represent them on a cladogram like this, well, we're going to see both lions and coyotes over here in this mammals kind of category, right? That would have to continue to branch there to represent those. So we have four limbs. They all walk on four limbs, which is kind of what distinguishes them, or at least one of those factors that distinguishes them from us. Felidae, again, have those retractable claws, right? Carnivora have shearing teeth. And so we can kind of distinguish them from there on, looking at some of those derived characters, those traits. So if we look here, here's clade carnivora, where we would have wolves and coyotes. Here's clade Felidae, where we would have lions and jaguars and leopards and all those other types of big cats. And so what's separating them is really this presence of these retractable claws, but both have those special shearing teeth because they're both in clade carnivora. But the Felidae grouping specifically involves just those larger cats, because these guys over here, dogs and their relatives like wolves and coyotes, don't have those retractable claws. Now, we could also go down the line and say, okay, well, both of these species share a category with marsupials, kangaroos. Both of these species share a category with reptiles, in that we're all amniotes, meaning that our young develop an amniotic sacs, an amniotic fluid, which is a little bit more advanced than what we see happening with amphibians and fish and other types of animals. But we do belong to the same group here because we do have four limbs, right? Not that we have to walk on all four, but all of these organisms here have four limbs. So categorizing these things, again, we're looking at physical features. We can even look at molecular information like DNA, and comparing those sequences together. But the bottom line is, again, the closer they are in these diagrams, the more likely they are related. So here we have new relationships that we can sometimes infer from, especially using things like molecular data. So what we've identified is we have a group of species that you all have heard of and probably, don't have a lot of harsh negative feelings towards, which are pandas. And pandas, for a long time, there's a whole category of pandas, or what we call pandas, that are actually red pandas. But if you've ever seen a red panda, they don't resemble the giant pandas that we see in East Asia. Red pandas are much more like raccoons or even squirrels or other organisms that we tend to have maybe even in the Smoky Mountains and around our community. They certainly don't seem to resemble polar bears or grizzly bears or any other type of bear-like species, but we do call them pandas. And so what's been identified through these types of cladistics, types of classifications, and doing this process based on evolutionary history, is that red pandas are much more closely related to things like raccoons than they are to any actual pandas or any other type of bears. What actually happened based on this diagram is we had a splitting event here, and then towards the left, we diverged and became raccoons and red pandas, and then towards the right is where you get those larger bear species. So we can get this information, obtain this, and organize this a little bit better, sometimes using cladistics, all right, to create those phylogenetic trees. So the last slide here, looking at our current tree of life, right? We're looking at the different categories of groups for all these. So we have color-coded on this diagram. You have the eubacteria, or the true bacteria here represented in blue. Those are thought to be the first grouping of species found on the planet and likely ancestors to everybody else in this line. Archaea here, remember, are significantly different from bacteria. Those are these extremophiles that can live in crazy hot or salty or oxygen-deprived environments. And so those guys follow here on this line. Obviously, these two types of prokaryotes would be more closely related to each other than they are to all these eukaryotes here. But when we think about the domain Eukarya, remember we have four different kingdoms there. We have Protista, which is represented by the yellowish color, fungi, which is the brown, Plantae, which is green, and Animalia over here on the right, which is red. And this is kind of deceiving because we really don't have that many more animals as compared to other things. However, they're trying to emphasize to you the different characteristics, right, of what makes each grouping different. So you see how these things kind of branch off, right? We have all these nodes going across the bottom, but then we have additional nodes here coming off of this. So where we have plant-like protists here, we have the divergence, we think, of all the plant species that exist coming off of that same branch here. So there'd be some type of derived character that they share here. Same with fungi, right? It seems that we're splitting here. We're taking one of two pathways, and my fungi are here, and my other protists are here. And continuing on that direction, you've got animals, and we've got even smaller nodes within those categories too. So again, the idea here with cladistics, not that it's in like contrary to Linnaeus's work, it's not. We can still apply that here, and you can see that with the vocabulary we're using. It's just in Linnaeus's work, we're looking at the specific categories of these things. And in cladistics and phylogenetics, like we're seeing here, we're looking at using those same categories, but relating them now to infer those evolutionary relationships, right? So we're kind of plotting them and organizing information in this case based on common ancestry, whereas Linnaeus's system really didn't take that into consideration. Okay. So that's all I've got for all of Chapter 9. There were only 2 sections. If you've got questions, let me know. Otherwise, we'll be reviewing and then testing pretty soon. All right. Bye, all.